I just want to start with some kind of reminder and announcements for this week. So those of you that have finished your lab exam, I have those graded, and if you want to come by and look at them the next couple of days, the best times would be tomorrow, Wednesday, 8 to 9, or after 1:30 up at my office if you want to come by and look at them. My plan is for people who can finish those this week, I'm going to use those grades as well as this next lecture exam grade in your midterm before I post midterm grades. So I do plan to get both of those exam grades finished and calculated into midterm grades. Remember that Thursday morning we have our next lecture exam, and I'm thinking right now that that's just going to be on paper in class. So if you're having to quarantine, I think that you can do that lecture exam at a later date, if that's okay with you. And then on Thursday afternoon, there will be a video instruction that I'm going to make for you all to get started with your paper project. And that video instruction will be describing how do you find the appropriate journal and start looking for articles that you want to use for your project. So I will have that video ready by whatever is your usual lab time on Thursday, and you will need to watch that and get started on your journal search instead of coming to class. And that's because I have to meet with Board of Trustees Thursday afternoon. So that's kind of our schedule coming up this week. And if you have any questions about it, or any questions generally, just you can ask during this session, or you can email, or let me know, because I will be around. I just won't be able to come to lab literally. I'm supposed to stay in my office and wait for the techs to join the meeting Thursday. So those are our announcements. And I wanted to let you all ask any questions that you have, or if you want me to go over certain things from this unit, we can do that this morning. And I think I have gone back and adjusted the quiz grades for this unit for the reading quizzes. I think I'll check that later this morning. But for those of you that maybe just joined after I started talking, if you want to come by and look at your lab exams, Wednesday, tomorrow is the best day to do that. So what kind of things do you all want to ask or would like me to go over this morning?
Dr. Ong.
Uh-huh.
Would you mind repeating the first part of the announcements? Jake and I came to the classroom. We didn't know it was on Teams today.
I'm sorry. Yeah, repeating the first part of the announcements. So I think I had first made the announcement about if you want to come see your lab exam. I have finished grading the ones that are completed, and the best time to do that would be tomorrow, Wednesday, between like 8 and 9, or after 1:30. I'll be around my office. The second announcement is that we have lecture exam Thursday morning, and that will be in class on paper. If anybody has to be in quarantine at that time, I would rather you put that off and come and take it on paper when you get out of quarantine. And then Thursday afternoon, we will not be meeting in person for lab, but I will have a instructional video to talk about how do you find your journal articles to use for your paper project this semester. So that video, you can watch it Thursday afternoon at your usual lab time, and it will probably be, this I didn't announce before, but it's probably going to be about 30 minutes of video, and then you'll have some time to actually search journals and find 2 articles that you're interested in. Then you'll submit those 2 articles to me, and I kind of look through them and tell you whether I think that one, which one would be the best. So that's kind of what's coming up this week. And any other, like this time that we have right here, the next half hour or so, any questions that you all have, you can go ahead and ask, or if there's certain things that you want me to review, I can do that as well. So sorry about the going to the classroom. I was going to send an update email and it just didn't happen. But
That's okay. I walked in and Jake was just there, and I looked around and I was like, oh no. So I got on Teams and we found it. So
okay, good.
That's okay. Thank you.
Yeah. And what I plan to do is I have all of the PowerPoints from this unit, and I can share my screen and show different things. So thanks for asking me to repeat. Let's see. I thought that I thought that I might kind of start with looking at the memorization tasks, and then go back and talk through some of the key figures. So I'm going to share my screen with you all. There we go. So remember that for this Thursday exam, this would be exam 2 for us. You want to know the directions of the ion gradients. So for sodium, potassium, calcium, and chloride, on which side of the cell membrane are those in greater concentration? And that also tells you which way are those gradients oriented. So if a channel opened up for one of these ions, you would be able to predict which direction it would move. And that's going to be really helpful for you all as all through your time in biology. This second one, knowing the steps of glycolysis. There's 10 steps, and for each step, it would be good to have memorized what is the product, what is the reactant, what is the enzyme, what is the product. And then remember that the product becomes the reactant or the substrate for the following step. So start with the names of those, and then once you've got the names, then you can work on structures of substrates and products. And that's going to be asked in the form of a big page-long table, and each of the 10 steps is a row in the table, and different things are left out in different positions. So you might need to draw a certain substrate on one line, but the next line might ask you what's the name of the enzyme that does this, and maybe the third line has name this product, and so on. Third set of items is know what are the locations, major products for Krebs cycle or citric acid cycle and electron transport chain. And the major enzymes, I think that's really just for electron transport chain. But know some of the key information about those 2 processes. And then the key figures, I need to get my textbook and look through these key figures. So what I'm going to do is kind of talk through these and remind you of important things. Anytime that you have a question, just unmute and go ahead and ask it. So I'm going to get my textbook real quick.
Hey, Dr. Ong,
Uh-huh.
are there going to be any figures given to us for this exam like the last one?
Yes. So one reason why I wanted to look through the key figures is so that I can remind myself what the majors are. So I know one of the figures is that that table of glycolysis, and that actually has quite a lot of info on it because it's basically telling all 10 steps with certain bits of info left out. So that is a whole page. One of the key figures that I know is there is going to be something like this. So something like this electron transport chain figure, except it's a different version. It's not this exact figure, but it is a similar type of figure showing all the components, but a different version. And so you have that to look at and then answer questions about. So that's one of the figures that will be on the exam. And then let me look through here in my textbook and remember what I've got listed for key figures. Okay, so it looks like it starts with 12.2. Let's see what that is. This exam is probably going to have more multiple choice on it than some than the previous exam. Oh yeah, 12.2. Okay, so that is the figure that talks about where or what substances can cross the membrane on their own or not, which is this. Let somebody back in the Teams. So figure 12.2, this figure is not on the exam, but you need to know off of this figure what are the molecules that can go straight through a membrane on their own. Basically, oxygen, CO2, water. And basically anything else besides those will have to have some kind of transporter to get through this membrane. That's not going to be a figure printed on the exam, but that's a set of facts that will help you do the exam. So let's see, 12.5. Okay, 12.5 is this concept. So the concept that any given molecule, when it's going to be able to pass through a membrane, it has a concentration gradient. So that's relying on how much of a molecule is one side versus the other. But there is also an electrical gradient. So remember that animal cells will normally have positive charge on the outer leaflet of the cell membrane and a negative charge on the inner leaflet. So any of those given ions, like these that you memorize, they have a concentration gradient that the cell builds up by using pumps to pump these to one side of the membrane. But depending on what their charge is, they also will have an electrical gradient that might help pull them across the membrane in one direction or prevent them from coming across in the other direction. So I think there are a few multiple choice type questions about predicting ion movements, like which way or which way is this part of the gradient pointing, which way is this part of the gradient pointing. So that's a useful concept to review. Let's see. 1211. Something else you will need to know is this basic information about sodium potassium pump. So for each ATP, there are 3 sodium moved outside, 2 potassium moved inside. And this helps set the directions of the sodium and potassium gradients in the first place, where we've got more sodium outside, less sodium inside. This figure is not on the exam, but the facts related to this figure you will need to know for this exam. 16. Oh yeah, okay, here's a figure that is, I think, actually on the exam. So I think this figure about glucose transport is one that I put the figure and then I ask you questions about how all this works. And I had forgotten that until just now, but this is a key figure, and I think I either print this on the exam or I put it up on the screen for you to look at and describe how glucose transport works. So remember that we've got co-transport on the apical side, where glucose kind of rides into the apical side of this gut lining cell along with sodium. So we've got a co-transporter here, and on this side there's a passive transporter just for glucose. And having that spatial arrangement is allowing glucose to get pulled from the gut into the cell down to the basical side towards the bloodstream. So you might want to make yourself a note that down here on the basal side of this cell, there is bloodstream, and that's where the glucose is generally moving toward. So this particular figure is, I think, printed on the exam, or I will have it up on the screen in the room. So I'm glad you asked this question. That was a good question to ask. Okay. 13.3. Okay, so those are probably most of the key concepts out of membranes and transporters. Okay, the next key figure is this one, which is just a really good overview. So this particular figure, it is not printed on the test and it's not provided on the test, but it's a good reminder of what's generally happening in glycolysis. And where is that happening? It's in the cytosol. You may want to write in here, if you haven't already, that there is a big complex that converts pyruvate to acetyl groups, and that's the pyruvate dehydrogenase. That would be acting right here. And if the way that I learn, I would use this figure to write on, there's citrate involved, there is oxaloacetate involved here, there's succinate involved, and also I would draw a little arrow coming off here that there is a GTP made in this step to kind of remind me what's happening generally during citric acid. And also draw in an FADH2, just to remind myself. And have both NADH and FADH2 dropping off electrons to electron transport. So when you're studying something like this, I think it's really useful to kind of mix and match, slides, if that makes sense. So this, where it's talking about NADH is going into oxidative phosphorylation. This NADH here and this NADH here are basically the same. And these two pictures are connected to each other. So when I feel like it's when you're studying for exams in my class, or really any other class, studying concepts as a holistic group is really helpful. So for me, taking this figure and then maybe putting other figures around it, like maybe putting this one over here, and then, you know, having a copy of the glycolysis steps and putting that next to this and making like a big spread out map of everything that's going on. For me, that's really helpful if I'm trying to think about a process with a lot of steps and how it all works together. Let's see what's next here. Whoa, oh, how did that happen? My book fell onto the space bar. Great. Okay. Let's see. What's next? I have 13.7 listed as a key figure, but I am not going to focus on that. And what that one is this. This is an important concept, but this is not going to be a key type of figure that you need to study for this exam. This is not going to become a big points question. And these steps are kind of basically listed anyway in your big glycolysis chart. So I think you could, for this semester, you could take that off of key figure list, that 13.7. The panel, this panel 13.1 is a 2-page spread of all the glycolysis steps, 1 through 10. So that is pretty useful to study, but it's basically this same stuff just listed on 2 pages of your book all together. So that panel is broken up in our slides into these steps boxes. That's what that is. Let's see, 13.12. Figure 13.12 is your citric acid cycle overview. Where's that? 13.12 is this. And again, you might want to highlight on this what are the important parts, which are basically anything that's being produced by this cycle. So anything that you see in the arrow coming out of citric acid cycle, that's important. And remember that this cycle would run one time for each acetyl group that comes into it. So remember, in glycolysis, we've got, go back to that. Oh, I've lost my place. Go back here. Okay. So remember that in glycolysis, we're getting 2 pyruvates out. Each one of those gets converted to an acetyl group, and each one of those goes through citric acid cycle, but there's 2 acetyl groups. So all of this that gets produced, it really would be 2 of those per glucose, because there are 2 acetyl groups coming into Krebs cycle, but it's individually. And remember, if you haven't done this yet, there is a chemical here in this region called succinate that you need to associate with citric acid cycle that's an important intermediate. So you might want to write that in if you haven't already, succinate. Okay, and then let's see, chapter 14. This table 14.1. Oh yeah, table 14.1. Find that slide set. It's this. There we go. Table 14.1 is this. So this where you have the accounting of how many ATP per output of glycolysis, and this is the intermediate step, and these are then made in citric acid cycle. So one thing that might be useful to write on here is, okay, both of these are coming from glycolysis. This NADH is coming from the intermediate step, the action of pyruvate dehydrogenase. And these outputs are coming from citric acid cycle. And those are things that, like, you probably already have memorized. What's new about this chart is this region over here where it's saying how many ATPs does that eventually become. And we have the total of 30 ATPs made during all of this aerobic respiration. It's not 36, like you may have learned in high school. I actually don't know what Dr. Vaughn is teaching in Gym Bio 1. I haven't checked that with her, but 30 is the accurate number of total ATP made per glucose. So this part is not new stuff to learn. You're already learning it when you learn about glycolysis in the intermediate step and citric acid, but this part is the key part of that table. And then the last, what is the last key figure? 1424. Oh yeah. Yeah, this is my favorite graph. Study this when you're studying this. So when you're remembering all of the components of electron transport chain and what order they go in, it's useful to study this picture together with this picture, because this is explaining why different electron transport chain components pass electrons specifically one to another, because they match in redox potential, and the place where they're passing electrons one to another also has a spatial aspect to it. So for example, cytochrome BC1 complex always passes electrons onto cytochrome C because they are spatially configured to do that, where cytochrome C drifts in the outer leaflet. And they also have a correct match of redox energies to make that transfer possible. So this is an important concept to study along with this picture together. So I hope that's helpful. I did record this. I am recording this currently, if you want to go back and watch it. And I am going to hop off now and attend my community board meeting. But if you have other questions about the exam, or as you're studying, if something is not making sense, just let me know that, and I will be checking email and get back with you. I'll also be in my office quite a bit tomorrow in person, so you could do a Teams chat with me or come by if that suits you. 8 to 9 is a good time, and after 1:30. So I am going to stop recording.